Hello students, myself Ramya, Assistant Professor and Head, Department of Biochemistry, SBRR Mahajana First Grade College, Mysuru. So yes students, today we will be discussing on one of the aspect of biochemical techniques, that is affinity chromatography. So before entering into affinity chromatography, yes, as a biochemist you all know that we are completely dealing with different kinds of biomolecules, right. So biomolecules might be either carbohydrates, proteins, lipids or nucleic acids. So studying all these biomolecules, obviously we require one of the major step, that is isolation. Yes. So these biomolecules has to be isolated for their studies, either with your application studies, either for their structural studies and characterization. So if this has to be isolated or purified, we require certain techniques that has to be employed for its isolation. So these techniques which has been involved in their separation, purification and isolation, we call it as biochemical techniques. So biochemical techniques includes varieties of techniques that is still more employed in every field, which includes centrifugation, chromatography, electrophoresis technique. Yes. So out of these techniques, today's classes we will be focusing on one of the aspect, that is chromatography. So which is one of the isolation technique which is employed for isolation of starting from basic biomolecules to all organic and inorganic biomolecules. So to begin with affinity chromatography, let us first focus on what is chromatographic technique. So almost in 1990s, this effort was put by one of the scientists, that is Michael Sweat, on where he separated the pigments, that is chlorophyll pigments and varieties of plant pigments, and isolated based on the principle of partition coefficient. So that led to a foundation of discovery of various kinds of chromatographic techniques, starting from your planar chromatographic technique till the advanced technique, that is UPLC, FPLC techniques. So what is this chromatography stands for? So as we already know that it's scan technology, it's application of techniques, and the chroma stands there as color. So initially the separation was based on the color, how they are getting distributed by using the principle of partition chromatography or partition principle or distribution coefficient. So based on the different migration rate, how the molecules are getting distributed between the two immiscible phases, the molecules were getting isolated or separated. So this stands as one of the major technique and leading to the separation of various biomolecules. So yes, under chromatographic technique, so you can define it as either as a biochemical technique or as biophysical technique, which includes the separation of mixture of components, and you can separate it as an individual components from a complex mixture, where each of the molecule will be migrating at a different rate. So either you can allow the sample, you can also call it as an analyte, to pass through a solution or a suspension. So due to their migration along with over the stationary phase, the molecules will get distributed or separated. So in generally, you can say this chromatographic technique as one of the physical methods which is used for the separation and distribution of those molecules over the stationary phase as well as with mobile phase, and get separated based on the different migration rate. So it depends on the type or nature of the mobile phases as well as with the stationary phases. So this chromatographic technique, to begin with, we need to know about two important terminologies, that is what is stationary phase and what is mobile phase. So stationary, the name itself indicates that it is completely fixed. So stationary phase is a supporting medium or the place where a molecule or an analyte is getting held up or hold on the surface of the stationary phase. And the other terminology is the mobile phase. So this mobile phase, the terms indicate as mobile, which means it acts as a carrier. So this is going to carry the sample or an analyte whenever you subject a molecule or a complex mixture for a chromatographic run. So this mobile phase, either it can be a liquid or either it can be a kind of gas. So any of these mobile phases, the role of this mobile phase is to carry or swipe away the sample over the stationary phase, where the analytes, either which prefer with your mobile phase or stationary phase, they get partition between the two immiscible phases. So this stationary phase, you can start from your paper, wherein the cellulose present in the matrix can act as a stationary phase. Starting from your paper, that is Watchman filter paper, till the next level of column chromatography, there is different kinds of stationary phase. You can include the packing of gel inside the column and at the planar level you can see the paper. So, this stationary phase sometimes you can directly use it as an application level, where for example, in paper chromatography the paper, that is Watsman filter paper, will act as a stationary phase. Whereas in column you can use the gel beads or ion exchange resins and you can pack the column. In some of the cases these stationary phase can be coated on a supporting medium, and you call that supporting medium as the solid surfaces, where the stationary phase either it can be coated or smeared on the surfaces. So, once we are clear with the what is mobile phase and stationary phase, to understand the next principle of affinity chromatography. Let us understand the classification of chromatography. A brief note or a brief overview on the classification of chromatography. So, this chromatography technique can be classified and divided based on various parameters. So, it can be based on geometry, it can be based on mode of operation, it can be based on nature of stationary phase and mobile phase and all. So, right now we will focus on the based on geometry, wherein based on geometry the chromatographic techniques are classified into two broad types. One is planar chromatography, another one is columnar chromatography. So, planar, as it indicates, one dimension wherein you are going to perform the chromatographic technique using the paper or TLC plates as in stationary phases. Whereas in case of columnar chromatography, the term indicates as column, wherein you are going to separate the analytes using a column as a supporting medium for the stationary phase to be packed. So, this columnar chromatography can be further classified based on the mechanism or the mode of separation. So, based on this mode of separation, we have many types of column chromatographic techniques. For example, you can look over the slide. We have size exclusion chromatography or gel filtration chromatography, wherein you are going to isolate based on the principle of size, and ion exchange chromatography, wherein you are going to isolate it based on the charge. And next, the topic of today's session, that is affinity chromatography, wherein you are going to isolate it based on the specificity or specific interaction between an analyte as well as with the stationary phase. Then reverse phase chromatography, HPLC, that is high performance liquid chromatography, UPLC, FPLC. So, out of all these techniques, today's concept or today's session, we will be discussing or focusing one of the types. So, you can look over the next slide, wherein it represents the paper, different kinds of chromatographic technique, TLC, columnar, and up to the advanced levels of different kinds of chromatographic techniques. And now, we will be focusing on one of the important columnar chromatographic technique, that is affinity chromatography. So, let us focus now. What is affinity chromatography? What is the principle behind the affinity chromatography, or how a molecule or an analyte can be separated or isolated using the affinity chromatographic technique? So, as the term indicates, affinity is a kind of attraction or a kind of binding affinity which has towards a specific molecules. So, this affinity chromatography is one of the type of columnar chromatographic technique where the mobile phase will be of liquid. So, you can also call it as affinity chromatography as a solid liquid type of chromatographic technique, where it is one of the most important powerful technique for the isolation of very specific biomolecules, including carbohydrates, proteins, nucleic acids, even hormones, receptors, antigens and antibodies. You can isolate it, and this mode of separation is for specifically for purifying the specific analytes or a specific molecules from a complex mixture. So, since it is based on the separation of specificity, you can say this affinity chromatography is one of the powerful technique which has high specificity and high resolution technique for the separations, because the phenomenon of separation is based on the specific interactions or specificity, which means the mode of separation in affinity chromatography will be the interaction between the biomolecule, that is the target of interest, which you need to isolate along with the stationary phase as well as with the mobile phase. So, this technique plays a very important role in isolating various biomolecules, as I mentioned. It also plays a very important role in immunology for the separation of antibodies, protein purifications, even in enzyme kinetics, the separation of specific enzymes. Yes. So, as we discussed with the general term as stationary phase and mobile phase, let us look into the stationary phase. So, what kind of stationary phase is used in case of affinity chromatography? So, in case of affinity chromatography, we use the stationary phase as the column which is being packed with the complete stationary phase, and that stationary phase it consists of gel matrix. the matrix, which is nothing but made up of packed with gel beads, which is completely swollen and it is packed with the gel beads. But prior to the packing, you need to modify the matrix. And what is that modification is? The gel matrix which is going to be packed in the column for performing an affinity chromatographic technique includes the involvement of ligands and the other term, that is spacer arm. So, gel matrix or the gel beads which is going to be packed here is linked or covalently connected with the ligands as well as with the spacer arm. So, let us look into the spacer arm and ligands in the upcoming slides. And the other phase, that is mobile phase, is nothing but your liquid phase. It can be any kind of organic solvents. So, when a molecule or an analyte or a mixture of samples is subjected for affinity chromatography, it gets distributed or partitioned between the stationary phase that as well as with your mobile phase. So, once we are clear about what is stationary phase and mobile phase, now let us see what is this kind of stationary phase, how the column is packed before understanding the principle of specificity. Now, we can look into the slide. We have a specific column and the column size, you can vary the size of column depending on the type of analytes and the volume of sample what you are going to take. So, now this is your column which is completely packed with the gel beads. So, a fraction of your column I am going to explain in terms of an individual gel beads. So, you can look into the slide. The place where you are going to see the gel beads contains the center ball like structure or a spherical like structure represents the gel matrix, and each of these gel matrix that is present over here will act as a stationary phase. And selecting this stationary phase is also very important. So, depending on what type of the sample you are going to isolate, depending on that you can choose a suitable gel matrix for your isolation purpose or the purification purpose. And this gel matrix is not as such packed into the column. This has to be covalently linked to another two terminologies or another two aspects, that is ligands as well as with spacer arm. So, gel beads which is going to be appearing in these gel beads, either you can use the cellulose, you can use dextrose, or you can use the other kinds of molecules which has high functional groups present on their surfaces. Even agarose can be used as your stationary phase or the gel beads for packing column in a affinity chromatographic technique. Now, you can see the surface of this gel beads which is present over here. It is surrounded by a huge antenna like structures and which is represented by blue color, and that antenna like structure especially which contains a crescent shaped structure, and that crescent shaped structure represents the ligands. Okay. So, each of these gel beads, I am showing an individual gel beads over the slide over here, but the entire column is being arranged in the same particular manner. So, this gel beads is being connected by a ligands, and this ligands is indirectly connected with another stalk like structure which is represented with blue color, that is what we call it as spacer arm. So, in general overall the stationary phase in an affinity chromatography includes the gel beads which is surrounded by or which is immobilized by the ligands via the spacer arms. Now, let us see what are these gel beads and how it is being packed to understand the principle of affinity chromatography. So, whenever we start or when it to begin with your affinity chromatography, obviously the column packing is one of the major step. So, this column has to be packed in the sense the gel beads are ready. Now, the next prior step is you need to immobilize or fix the ligands on the gel matrix. So, obviously any kind of molecule, if it has to interact or bind with the molecule, it has to require or it finds a certain recognition sites or any kind of active sites to bind. So, obviously what the material we are going to pack as gel beads, these gel beads will also have the surface functional groups where actually these kind of ligands are going to bind. So, the ligands are those molecules which are going to bind to the surface of your gel beads. So, these gel beads should be suitably selected, wherein you have to look in that these gel beads should be highly soluble in your solvent whichever you are preferring for the separation. They should be chemically, mechanically inert in nature, because if they are not inert in nature, as soon as you add your analytes, it can damage your sample or a interest what you are going to be isolated. So, you need to choose such a matrix or a gel beads. It should be chemically and mechanically stable, and also it should be easily or covalently link easily with your ligands for the separation, and it should have very good flow properties, and the most important thing is it should have the large surface area. So, higher the surface area, larger the more the amount of ligands that is going to bound, and more the number of molecules are getting isolated. So, look into these properties and prepare your gel beads. So, once the gel beads has been done, the next thing is you need to fix with the ligands, and you call that step as immobilization of ligands. So, this immobilization is nothing but the fixing or attachment or covalent binding of these ligands on the surface. But prior to binding of these ligands, the thing is the gel beads whichever you have been prepared, it has to be priorly activated. You need to pre-activate the gel beads. The pre-activation includes either you can treat the gel beads with any of their alkaline solutions or cyanide bromide solutions of alkaline pH. So, this activation includes the surface activation where the functional groups which is present on the surface of the gel beads, they get activated or they get exposed, wherein they provide the site for binding of ligands. So, you can look over the slide, we have a gel matrix, gel beads. So, on treating with cyanide bromide, you can see the activated functional groups which is represented by the yellow color. So, once the functional group is being activated, the next thing is you are going to prepare or allow the binding of ligands, what we call it as this immobilization of ligands. So, whenever you choose your ligands, even you need to take a precaution that as soon as you choose your sample to be separated, you also have to design or select the type of ligands you are going to prefer. Suppose if it is an hormone receptor concept, you can specifically you can use as a ligand as an hormone, or if it is an antigen antibody, you can use an antigen as a ligand or an antibody as a ligand. So, specifically choose the ligands depending on the type of sample of interest to be isolated, and most of the ligands they are going to bind covalently on the functional groups. So, these ligands are nothing but the molecules that reversibly binds to the matrix functional group. Why it is said as reversible is because after isolation they are the one which is going to hold the sample during the migration of the analytes or a mixture of sample. So, as soon as it is held, the molecule has to be obtained back, right? It has to be retrieved back after the isolation. So, the binding has to be detached from the ligands. So, we call this process as biological or reversible binding of the molecule. So, this reversible binding property of ligands helps for the isolation or purification of the samples. So, once the covalent linkage has been done by your ligands, you can look over the slide wherein the functional groups, that is the yellow colored part of your gel matrix, is being isolated or it has been activated, which is being done by cyanide bromide solution, is being occupied by ligands via the covalent linkage. So, whenever you are choosing the ligands, as I already mentioned, you can look into the slide over there wherein you have the ligands, and the exposed part represents the outward protruded part represents the actual binding site where your target molecule will covalently come and bind. So, during your chromatographic run. So, when you are performing your binding or the immobilization of the ligands, even this ligands has a capacity or you have to choose a suitable ligands for binding it. Suppose if it is irrelevant ligands, it may also damage or disrupt the surface of a matrix. So, once the matrix or the gel bead surface is being disturbed, even the functional groups or an active sites which is present on the surface get denatured. So, once it denatures, it loses the affinity even though if the target molecule bypasses. So, suitable ligands and binding of suitable ligands is also very important before performing your affinity chromatography. So, once your gel backing is done, you have to fix with your ligands, we call it as immobilization. So, once the immobilization is done, earlier once the molecules are attached directly with the help of ligands, there was another problem is, you can look into the slide wherein there is a complete steric hindrance. You can look over the side, we have a gel beads, on the gel beads if the ligands are attached, and when the target or a mixture of molecules is allowed to pass through the column, as the analytes migrates to the column, they are going to bind to the surface of the ligands. So, as they bind, you can see the overlapping of the molecules. So, the yellow one represents the targeted molecules, that is what the molecules you need to isolate it, which has a specific affinity for the particular ligand which is immobilized on the gel matrix. So, as it passes, we can look over the slide wherein it is going to isolated, the molecule or the target molecule, it is going to overlap on your gel matrix. So, this overlapping of the targeting molecule between the molecule of ligand as well as with your bead surfaces may prevent the binding of your target molecule with a complete access. So, they do not get a complete access for binding a molecule. So, in such cases, it creates a kind of steric hindrance. This problem, which is being analyzed, is further modified by the use or the replacement with a spacer arm. So, now let us look into that. You can look into the picture, wherein initially the ligand is just the molecule with an hexagonal structure, wherein now you had provided with a stalk like structure which acts as a bridge or a short gap like structure between the gel bead surfaces as well as the ligands, and this stalk like structure you call it as the stationary phase, and this stationary phase is being connected via a bridge called as spacer arm. So, this spacer arm is one of the important part of your gel matrix preparation, wherein it is going to avoid the steric hindrance and provide the complete access of your molecule of interest to be separated to completely bind with your ligands. So, this spacer arm, many spacer arms that has been used, and using this spacer arm as a role to prevent the steric hindrance also has to be selected in a suitable manner. For example, selecting a spacer arm is also very important. You can, some of the spacer arms that has been so far used, you can look into the slide, that is hexamethylene 6 aminohexanoic acid. So, in all these examples, whatever we are quoting, it all contains hexa, which means the spacer arm which is being used for immobilizing the ligands or bridging the ligand between the ligands as well as with the spacer, that is the gel bead surface, is around 6 carbon chain length. So, the spacer arm that is connected between the it consists of 6 carbon atoms, and approximately it may contain around 4 to 6 centimeter in length. So, what is the role of the spacer arm? Now, we can look into the slide. Rather than the complete inaccessibility of your targeted molecule to the gel beads is now accessible freely without any steric hindrance by the application or by the use of this spacer arm. So, usually this spacer arm selection is also very important, as I already mentioned it. You have to choose in such a manner, it should be maximum length is around 4 to 6 centimeter. In case if there is a very longer spacer arm or very shorter spacer arm, that also impact on your purification process. Very longer the spacer arm, the length of the spacer arm, then it occupies more of the gel beads, where the analyte or number of molecules that is being separated reduces. Whereas, very shorter the spacer arm if you are going to use, then the spacer arm will again create a steric hindrance between the ligands. So, choose an appropriate spacer arms when you are going to fix it or immobilize the ligands. Now, this ligands can directly connect through the spacer arm to your gel beads, so that no steric hindrance and your molecule of analyte can have a good affinity for your ligand and can bind covalently to the ligand with a correct procedure. In general, the spacer arm can be around approximately around 1000 Daltons. So, this, when you are choosing a spacer arm, it should possess one of the characteristic. That characteristic is, each of these spacer arm which you are going to use for the isolation or purification should possess 2 functional groups at one terminal and the other terminal. The one terminal containing functional group, the group should be in such a manner, it should have an affinity for binding to the surface of the gel matrix. The other end of your spacer arm should possess a functional group which has a capacity or affinity for binding with the ligand, so that they can bridge between your ligand as well as with the spacer arm. So, this is how we are going to connect or prepare the stationary phase and immobilize it using the ligands. So, now once your gel beads immobilized with the ligands connected with the spacer arm has been done, you pack your column. Now, let us look into the complete principle of or working principle of your affinity chromatography. So, as I already mentioned, affinity chromatography is one of the liquid chromatography which specifically works based on the specific interactions. So, it works on the based on specific binding of a target molecule or an analyte of interest what you need to separate or isolate to binding a ligand, wherein it utilizes the reversible binding of ligand through via the affinity. So, specific interacted molecules will bind to the ligands and they retain in the column. So, once they lose the affinity or once they lack the affinity, or the molecules which do not have affinity with the specific ligands, they elute out of the column. So, elute is nothing but passing out of the column. So, based on that you can analyze the molecules that the compounds or analyte of mixture, when you subject it for affinity chromatography, as they traverse through the column or as they migrate to the column, the compounds that have very high specificity or with your ligands, they are going to bind with the ligands and they remain in the column for a longer duration, unless and until you elute the molecules or separate the molecules from that bounded position later. And those molecules are a mixture of samples which lack the affinity for binding with your ligands, they automatically while traversing they elute out of the column, which is carried by your mobile phase, which means the mobile phase will carry the sample and elute out through the column. Now, let us see into how these molecules are separated. You can look into the slide. We have gel beads, and these gel beads we have connected with the spacer arm, a stalk like structure, and a crescent shape, that is ligand. And what is that yellow cubes like structures that is being occupied at the position of ligands is, they are nothing but the molecules of specific interest. So, the red cube like structures, they have the specific interaction with the molecules where they are going to bound with the active position of the ligands. Where the other molecules which is of green, pink and blue, which is being represented, they are eluted out of the column. So, in general, affinity chromatography works based on the principle of specific interaction of between the mobile phase as well as with the stationary phase, which is having the affinity to stationary phase will remain bound in the column, whereas those molecules which do not have affinity will elute out along with the mobile phase. Let us see the overview of the affinity chromatography. So, I have taken one of the gel beads over here. We have a gel bead and the red tag label one represents specific ligand which is connected via the spacer arm. So, once the matrix along the gel beads or the immobilization of the ligands to the gel beads is done, you are going to pack the column with your ligands and spacer arm. Once the column is packed, you are going to take your sample of interest to be isolated or purified, and you are going to pour it, or you can tell it in terms of loading of sample. You can load the sample of mixture of sample to be isolated into the column. As the column traverses, now you can look into the slide, we have a mixture wherein it is represented by different colors like orange, yellow and blue color. So, as they traverse through the column, so one of the specific molecules which can identify the specific functional group on the ligand, they are going to bound. For example, in the slide which has been presented, we have a specific molecule that is which is represented by a orange geometrical figure, which has a specificity for binding a ligand. It binds and remain in the column. And this second phase of chromatographic technique, that is affinity chromatography, which includes binding of targeted molecules. So, even initially during the immobilization of your ligands, as the gel beads is being immobilized with ligands, you also have to allow certain duration of time, what we call it as incubation time, for binding the ligands into the gel matrix. After certain incubation of time, you wash out the unbound ligands. Then you can reload the sample to the column. Once the sample is loaded, the binding affinity exists between the ligands and the target of or the interested molecule to be isolated. So, once the binding of the molecule has been done, the other molecules which lacks the affinity, they are eluted out. You can see the other molecules which has been washed out. Now, how to isolate the molecule? Yes, we know that the molecules, here it has been completely bounded to the specific molecule or the specific ligand. These molecules can be further isolated by a process, and you call that process as elution. So, elution is a process where the bound molecule which is remained or retained in the column can be eluted out of the column. And this process where the target molecules which is bound or remain can be isolated or removed through your specific process, and you call that process as, or the phenomenon as, elution process. So, this elution, in case of chromatographic technique, we have two types of elution. One is non-specific elution and specific elution. So, obviously isolation includes that we need to isolate the specific target molecules, or you can collect it in terms of a fraction. If it has to be collected in terms of fraction, it has to get detached from the bound form to its and reaches to its unbound form, so that it can be elute out of the column, wherein we are going to collect it as a fraction. To elute those bound molecules, we have specific terms, that is elution process, non-specific elution as well as with specific elution. So, since the name says as non-specific and specific, let us see what is the first mode of elution process, that is non-specific mode of elution. So, this non-specificity includes that you are not going to use any kind of molecules which is having more specificity than the previously bound analyte or a targeted molecule. Here in this process of elution or method of elution, you can look into the slide. We have a procedure wherein we have already performed an affinity chromatographic where the molecule is already immobilized on the surface of your gel matrix. So, once the molecule is isolated, that is purified, leaving all the unbound molecules, now the thing is you need to separate the bound molecules by subjecting for non-specific elution, which includes non-specific elution. You can either perform it by treating the gel matrix or by treating the column through any kind of buffer solutions or the solution which contains a varied pH. You can alter the pH or the ionic strength of the column. Suppose, for example, if the solution or the buffer which has been prepared earlier for packing the column, if it is the buffer pH of 7, while elution or eluting process, you can use a buffer solution of pH 7. So, once you alter the pH of the solution and when you load it into the column, as the buffer passes through or traverses through the column, immediately the pH of the buffer alters the functional group surfaces. As the functional group surfaces get ionized, they loses the charges. Once they loses the charges, automatically the affinity between the bound molecule or the ligand get detached. So, as a result, the molecules automatically they get eluted out due to dissociation of targeted molecule with the ligand due to variations in the pH of buffer. So, this type of elution, whereby using a different ionic strength buffer or pH or buffer which is containing an alternating pH, you can isolate these molecules. So, you can look into the slide. The first picture represents wherein the bound molecules, the green color represents the targeted molecule of interest which is already bound. Now, you can treat this column with the buffer solutions or any ionic strength different pH of solutions which is not used for the preparation of gel matrix. As soon as you treat the buffer solution of varying pH, the pH of the buffer varies the surface of the charges and automatically it leads to the dissociation of these molecules from the ligands. You can see the green colored cube like structures, that is the targeted molecules, they are eluting out of the column. So, this kind or mode of separation we call it as non-specific elution. So, that is non-specific elution is performed either by altering the pH or by ionic strength. Suppose if you are using any kind of lipids or any hydrophobic samples, you can use a specific detergents or you can use a combination of solutions. For example, you can use 50% of ethylene and 50% of glycol. So, 1:1 ratio, you can prepare the solutions which allows the even hydrophobic molecules to get detached from the molecule as they lack the charges. In case non-specific elution, you might have experienced or you might see that there are certain contaminants. As you are altering the pH of the buffer solutions, you may get precipitates or you may get contaminants. So, further the elution or the fraction what you had collected after the chromatographic process, you can collect it and you can subject it for purification either by dialysis process, wherein you obtain the complete purified form of your targeted molecules. This is one mode of elution process via the non-specific elution. Let us look into the second process of elution, that is specific elution. So, the term indicates as specificity, which means you are going to use the specific molecules which can bind to the ligands, but make sure that the molecules which are going to treat with the ligands or the treat with the column already bound with the ligands has to be having the higher specificity or high competitive interaction than the already previously bound ligands. So, this mode of elution we call it as specific elution, which means in this process you are going to treat the molecules so that it creates a competition between already existed molecule and the molecule of interest what you are going to isolate it. So, once you are going to add the specific molecules which has high affinity than already the previously added ligands. So, you can look over the slide wherein the first picture represents again the same, the bound ligands after the chromatographic run, wherein the molecules which has affinity has bounded to the ligands. Now, you can treat it with any kind of molecules or specific molecules which has high affinity containing molecules than the previously added ligands. Suppose if you are using enzymes, you can use the enzymes which has similar functions and has high affinity than the previously added enzyme for the particular same substrate. Similarly, you can look into the slide there. I have taken a blue colored the geometrical figures as the other molecules which has high affinity than compared to the green color one. So, as you treat with the specific molecules on treating, as they travel or migrate towards the column, these molecules will replace the already bound molecules. So, you can collect the bound molecules initially, that can be collected as a fraction. So, this kind or mode of separation of molecules by using a specific very competitive or high affinity containing molecules, you call it as specific elution. So, thus while using affinity chromatography, you can either perform either with specific elution or non-specific elution. Thus, the molecules of interest can be isolated. Let us summarize now. When you are performing in affinity chromatography, we need to start in the sequential process. That is, first thing is you need to pack the column, we call it as the column packing procedure, and the content which is being filled, we call it as the column bed. So, once the column has been packed, the next thing is you need to add, wherein you are going to add the sample inside the column. We call it in terms of loading of sample. As the sample is loaded, as the molecule or the mixture of analyte migrate, they get separated based on their affinity. Once they get separated, the molecule is, we call it as that is binding or affinity containing molecules will remain held or bound to the ligands, and the other molecules are washed out. The next process is that you need to wash it and remove the unbound molecules, followed by you can perform the elution process, wherein you can perform either by specific or non-specific type of elutions. So, this technique, as we already discussed the principle, how a molecule which is having a specific interaction can be isolated by using this technique. So, not only your specific kind of proteins, this technique or has an advanced application level at different moleculars level. It can be used for the separation of very active biomolecules even in the functional state as well as with a denatured state. You can isolate and purify various substances in a larger volumes also. So, for examples, you can isolate with your antigen and antibody, you can isolate the enzymes, you can isolate either through based on enzyme substrate interaction, as you know that enzyme and substrate, the key lock theory, how they have an affinity towards binding a substrate. You can also isolate enzyme specific inhibitors by using a specific inhibitors and study what kind of inhibitors are specifically act on the particular type of enzyme. You can also take hormone receptor concepts. So, let me have a brief explanation on one of the example wherein you can apply the principle of affinity chromatography. That is, let us take an example. This is one of the slide which is representing how a molecule, that is target of interest, can be isolated, where the target of interest here will speak in terms of isolation of enzymes by using a substrate. So, you can look into the matrix. It has been connected with a spring-like structure that is represented by spacer arm, and the purple one represents the ligand which is immobilized on the matrix. Now, you can treat the enzyme which is present over here. The immobilized ligand which is acting, the purple color one which is represented, will act as a substrate. So, this is the gel matrix. You are going to load it in the substrate, that is the column. So, once the substrate has been loaded, the next thing you are going to load with the enzymes in a different kinds of enzymes. You are going to load it in a column. So, once the enzymes start traversing along with your mobile phase, this enzyme specifically isolates or specifically identifies the ligand and going to bind the ligand, and we call this as affinity interaction or biological reversible interaction between the ligand as well as with the substrate, that is the substrate and the enzyme, and form a complex. So, now after this binding of enzymes with a specific ligands, the enzymes which do not have an affinity for the specific active site, the active site on the enzymes do not have any active sites for binding a substrate, they are eluted out of the column, which means they are washed out of the column. Now, only the bound enzymes which is being present, you need to isolate it, wherein you can either subject for your specific elution or non-specific elution. This specific elution, as I already mentioned in the previous slides, you can either perform it by using any kind of enzymes which is having higher affinity to the substrate, to the same substrate what you are already bound, okay, or else you can perform by the non-specific elution by altering the pH. Once you alter the pH of the buffer and load into the column, the pH alterations will lax makes the dissociation of enzymes from the substrate, and you are going to elute out of the column. So, likewise we have another example wherein you can also purify with the proteins, not only the enzymes and substrate compositions, you can also subject it for protein purification, wherein you can use the multiple mixtures of proteins which is having specificity will bound. Here in this case, the targeted protein, which is represented in the grey color, will bound over it, and at the end of the process we have an elution wherein all the molecules which is having the specificity are eluted. So, thus a protein can be separated by using this affinity chromatography. Not only the protein or an hormone or a kind of enzyme substrate reactions, even various macromolecules can be separated by using your affinity chromatography technique. So, some of the examples I have quoted here, wherein you can separate avidin, a biocytin as a ligand, you can also separate alpha chymotrypsin, you can separate thrombin, and one of the major impact, that is on nucleic acids, you can also separate mRNA from the mixture of samples, wherein we already know that this mRNA is a single strand, also contains a poly A tail at your 3' end. If that acts as a ligand, then you can specifically use ligands that can be acting as a poly T. So, when you can fix the ligand as poly U or poly T, and you can load the mixture of mRNA samples, wherein the poly A mRNA which is containing in the mRNA will going to target or bind as a complementary base pairing with your the complementary bases which is acting as a ligand. So, in thereby cases you can also isolate the mRNA from your mixture of nucleic acids or mixture of mRNAs. So, thus not only in your protein, it is applicable in all kinds of fields. Yes, let us see or focus some of the applications as we already discussed. The principle of affinity chromatography, as we already discussed, how it separates based on specific interactions. They are also applied for purification and concentrating an enzyme solution, and you can also separate various kinds of RNA, complementary dRNA, purifications of mRNA, purifications of enzymes, immunoglobulins, vitamins, based on even the hormones and kind of drugs and some of the organic and inorganic molecules. Thus, we complete with the principle of affinity chromatography. Thank you.